Hello, my name is Immo Hansen from the Molecular Vector Physiology Laboratory at New Mexico State University. The mosquito fat body culture system we will demonstrate today is an in vitro organ culture system that we use in our laboratory to study physiology and metabolism of live fat body tissue attached to the abdominal body walls of mosquitoes. Insect fat body is an organ in which many metabolic processes take place. Its functions mirror those of liver and fat tissues and vertebrates. We have used this system to study the regulation of yolk protein production and the mechanisms of nutrient uptake in the yellow fever mosquito Aedes aegypti. Aedes aegypti is a principal vector of arboviruses like dengue, chikungunya, and Zika. The mosquito fat body is the sole source of yolk proteins in mosquitoes.
This video protocol demonstrates an in vitro mosquito fat body culture system and shows several experimental applications of the system. The protocol that we will be demonstrating is set up in multiple parts. First, we will be preparing the solutions and reagents necessary for the fat body culture system. Second, we will perform mosquito dissections to isolate fat bodies. And lastly, we will transfer isolated fat bodies to an amino acid-enriched medium to activate yolk protein synthesis. There are several downstream applications that can be performed following fat body culture. Today, we will be performing fat body culture followed by next-generation sequencing. We'll compare two samples, an unstimulated sample with one sample stimulated with amino acids and ecdysone. Additional materials and equipment that will be needed include an aspirator with collection vial for mosquito collection, CO2 fly pad hooked up to a CO2 gas bottle, ethanol to clean the fly pad, stereo microscope with light source, dissection micro scissors, fine tip tweezers, and 96